Chapter 5: The Fundamental Unit of Life. What are living organisms made up of?
We observe small buildings, bungalows and tall towers, which are the places of human residence. These are the constructions using the same type of materials such as iron rods, bricks, cement, sand, stones, etc. These non-living things are made up of building blocks. That is the foundation which supports pillars, beams and slab. We come across walls, doors, windows of different size and shapes. Altogether, the structure is a cement-concrete construction having an external appearance as well as distinct internal structure. We observe a similar situation with living organisms. There are different kinds of living organisms on this earth. They differ in their size and shape. They live in different habitats, air, water, or land. All these living organisms are made up of the same building blocks. The common basic structural and functional unit of living beings is called a cell. A cell has a distinct structure as well as a specific function. In the year 1665, Robert Hooke discovered the cells in a thin section of cork, that is, bark of a tree, which is an outer protective layer of the trunk. He observed these cells with a self-designed microscope. The structures resembled the compartments of a honeycomb. The compartments were empty. The cells of cork were dead and without any content. He labeled the compartments as cells. The cell in Latin means small room. Every living thing may be different from the other, but it is made up of separate tiny units called cells.
Let us understand these small units with the help of an activity.
Activity. Take a small piece of onion bulb. With the help of pair of forceps, peel off the skin called epidermis from concave inner side of onion. Put it in a watch glass containing water. This will prevent folding and drying of separated peel. Take a clean glass slide. Put a drop of water on it. Transfer a small piece of peel on the slide with the help of a brush. Stretch the peel and make it flat on the side. Put a drop of dilute iodine solution on the peel. Place the cover slip carefully with the help of the mounting needle. Do not allow air bubbles to enter on the slide. This is a temporary mounting of onion peel. Observe the peel under compound microscope. Focus the slide first under low power and then turn it on high power. We observe rectangular structures attached to each other.
Try and draw these structures on a paper. Repeat the same experiment with different sizes of onion bulbs. What do you observe? You would observe that all the different size of onion bulbs show similar basic structures. These small structures are the building units for an onion bulb. Similarly, all living organisms are made up of building units called cells. As learnt, cells were first discovered by Robert Hooke. Later, in 1674, Leeuwenhoek discovered free living cells in the sample of pond water with the help of an improved microscope. In the year 1831, Robert Brown, a school teacher, continued the work of Robert Hooke on microscopes and invented an advanced microscope. He observed that there is a new world within a cell. He said that the cell was like a small factory with around 12 to 13 cell organelles within, each organelle carrying out a particular task. He discovered the presence of the nucleus, a spherical body in cells. In 1839, a Czech animal physiologist, Johannes Parkinge, named the living fluid present within the cell as protoplasm. Proto means first and plasm means colloid.
M. J. Sheldon and Theodore Schwann, 1838 to 39, proposed the cell theory. According to this theory, all plants are composed of cells. The cell is the basic unit of life. In 1885, R. Virchow modified this theory, saying that all the cells arise from existing cells. New cells are formed by the division of the parent cell into two daughter cells.
Invention of magnifying lens led to the discovery of many microscopic organisms. It was then known that a single cell constitutes a whole organism within itself. These organisms are called unicellular organisms. Uni means single. Amoeba, Paramecium, Chlamydomonas and bacteria are some examples of unicellular organisms. Whereas many cells group together in a single body to perform different functions to form various body parts in a single organism. These types of organisms are called multicellular organisms. Multi means many. Thus, organisms consisting of many cells of similar or different types performing different types of functions are multicellular organisms. Hence, it can be stated that each multicellular organism comes from a single cell. This proves the cell theory that all cells arise from the pre-existing cells.
With the discovery of electron microscope in 1940, it was easy to understand the complex structure of a cell and its organelles. The electron microscope has a much higher resolution power than a normal laboratory compound microscope. It magnifies and resolves the objects clearly.
Let us perform another activity to understand the variety of cells found in nature. Take different sizes of onions and prepare temporary mounds of leaf peels, root tips or peels of onions. What do you observe? You would observe that the basic structure of all the cells is the same, whereas some organisms have cells of different kinds. The shape and size of the cells are different. These different characteristics help the cells to function specifically on any organ. For example, amoeba has the ability to change its form. This is because the cells of amoeba are flexible and help it to change its form. On the other hand, some cells have a fixed shape and a particular function to accomplish. For example, nerve cells of a human has a typical shape and a particular function. Its function is to carry signals from brain to different parts of the body and vice versa. Cells have the capacity to perform certain basic functions. This capacity of a cell to function efficiently is a characteristic of all living things. As humans are multicellular organisms, each cell has to coordinate with the other cells to work properly. This coordination can also be stated as division of labor of cells in multicellular organisms.
In our body, the stomach is concerned with churning of food and helps in its digestion. The kidney is a chief excretory organ which reabsorbs the necessary materials and throws out unwanted waste products. The heart functions like a pump and circulates the blood through the blood vessels throughout our body. The ears are the external organs which are concerned with hearing of sound. Division of labor is seen within a single cell. Each cell contains some components which perform a specific function within the cell environment. The different components of a cell are known as cell organelles. Exchange of materials, synthesis of proteins, and release of energy are some of the specialized functions of the cell.
These functions are performed by specialized cell organelles. The different organelles constitute together to form the basic unit called the cell. A cell can survive and perform all its functions because of these organelles. All living organisms are made up of small units called cells. These cells have cell organelles which perform a specific function. The organelles of cell can differ from organism to organism, but their functions do not change.
Let's do a quick check. Let's do a quick check. Structural organization of a cell.
As we all know, a cell consists of specialized components called cell organelles. These organelles help the cell to live and perform functions efficiently. Each cell has its cell organelles organized in a particular manner. These different types of organelles work in coordination with each other. Different types of cells show special differences, but they all show a basic structural plan. This basic plan is expressed in a generalized cell. A generalized cell has three essential parts: a cell membrane or a plasma membrane, cytoplasm, and a nucleus. In addition to these essential parts, there are other cell organelles which help in carrying out all activities inside the cell. This structural organization of cells help in the interaction of cell with the environment. This interaction helps an organism to function properly. Let us study about the different cell components and their functions.
Plasma membrane or cell membrane.
The outermost covering of the cell that separates the contents of the cell from its external environment is called plasma membrane or cell membrane. Plasma membrane or cell membrane has tiny pores through which substances enter and leave the cell. The plasma membrane permits the entry and exit of some materials in and out of the cell. It prevents entry of some materials. Hence, it is called a selectively permeable membrane. Have you ever wondered how does the movement of particles take place into or outside the cell? Substances like carbon dioxide or oxygen move across the cell membrane through the process of diffusion. The spontaneous movement of substances from a region of higher concentration to a region of lower concentration is called diffusion. Diffusion plays an important role in gaseous exchange between the cells and also between the cell and its external environment. When carbon dioxide with high concentration accumulates inside the cell in more amount, it needs to be excreted out. The carbon dioxide with high concentration cannot move out of the cell unless the external environment of the cell has low concentration of carbon dioxide. When there is a change in the concentration, carbon dioxide moves out from the cell to a region of lower concentration. Similarly, oxygen too enters inside the cell by the process of diffusion when the concentration of oxygen is low inside the cell. As there is continuous diffusion of gaseous particles in and out of the cell, water too enters and leaves the cell through the process of osmosis. The spontaneous movement of water across a semi-permeable membrane is affected by the amount of substance dissolved in water. It occurs due to differences in the concentration of the solutes on either side. The spontaneous passage of water molecules from a region of high water concentration through a selectively permeable membrane to a region of low water concentration is called osmosis. Let us understand the process of osmosis by placing a cell into a solution of sugar or salt in water. When a cell is placed into a solution of sugar or salt, there are three possible situations. When the medium surrounding the cell has higher water concentration than that of the cell, that is, the outside solution is very dilute. Such a solution is called hypotonic solution. In such cases, the cell is likely to swell up. This happens because more water enters the cell and less water is released out from the cell. When the medium surrounding the cell has the same water concentration as that of the cell, such a solution is called isotonic solution. In such a case, water travels through the cell membrane in both directions, but the amount of water entering inside is the same as the amount of water exiting from the cell. Thus, it can be said that there is no overall movement of water and no significant change in the size of the cell can be observed. When the medium surrounding the cell has lower water concentration than that of the cell, the medium, that is, the outside solution, is more concentrated. Such a solution is called hypertonic solution. In such cases, the cell shrinks. This happens because more water leaves the cell as the outside medium is concentrated and less water enters the cell. Thus, it can be said that osmosis in a selectively permeable membrane is a special case of diffusion. Let us understand the process of osmosis with the help of an activity.
Activity: Osmosis with an egg. Remove the shell of an egg by dissolving it in 40% dilute hydrochloric acid. The shell is made up of calcium carbonate, hence dissolves in the acid. A thin outer skin encloses the egg. This is the plasma membrane. When we put the egg in pure water and observe it after some time, we observe that the egg swells. When we put deshelled egg in salt solution and observe it after some time, we observe that the egg shrinks. When egg is placed in a hypotonic solution, water enters the egg cell. Water is hypotonic to egg. That is why it enters the egg and causes it to swell. When egg is placed in a hypertonic solution, water leaves the egg cell. The salt solution is hypertonic to egg, hence water goes out causing shrinkage of the egg cytoplasm.
Let us perform another activity with dried raisins to understand the process of osmosis.
When we put raisins in clean water for a few hours, we observe that they swell due to absorption of water. The raisins swell because they absorb water from the outside environment. When we put the swollen raisins into a concentrated salt solution, they become flaccid, that is, they shrink. Water from the swollen raisins comes out and hence they shrink. Water is the substance which moves in and out of the raisins.
Unicellular freshwater organisms and most plant cells gain water through osmosis. Absorption of water by roots of the plants is an example of osmosis. Diffusion helps in exchange of gases and water in the cell. Cells obtain nutrients from the external environment. Different materials move in and out of the cell. We can thus say that plasma membrane is flexible. It is made up of organic substances called lipids and proteins. Due to the flexibility of cell membrane, unicellular organisms like amoeba are able to engulf the food and other material from its environment. Such process is called endocytosis. Structure of plasma membrane can be observed only under an electron microscope. With the invention of electron microscopy, it has become possible to examine and study precisely the complex ultrastructure of the cell and its very minute organelles in recent years.
Cell wall. In addition to plasma membrane, plant cells have another rigid outer covering called the cell wall. Cell wall is situated just outside the plasma membrane. It is non-living and freely permeable, which allows all substances to enter and leave the cell. It is made up of cellulose, which is a type of carbohydrate. It gives protection to the cell. It determines the shape of a cell.
When a living plant cell loses water through the process of osmosis, the contents of the cells either contract or shrink and move away from the cell wall. This phenomenon of contracting or shrinking of cell contents inside the cell wall is called plasmolysis. Let us understand the phenomenon of plasmolysis with the help of an activity. Activity. Let us understand the process of plasmolysis by using Tradescantia leaf, Rio leaf. Mount the peel of Tradescantia leaf in water on a slide. Put the slide under high power of microscope and observe the leaf cell. During observation, small green granules are noticed in the cell. These green granules are called chloroplast. They contain green substances called chlorophyll. Now put concentrated sugar solution or salt solution on the peel of the leaf and wait for some time. Water comes out of the leaf cell due to osmosis. Now observe it under a microscope. During observation, it is noticed that cytoplasm of the cell separates from the cell wall. What is the reason behind this? The solution outside the leaf cell is hypertonic. Therefore, water comes out of the leaf cells, which results in the cytoplasm of the cell separating from the cell wall. Now place some Tradescantia leaves in boiling water for a few minutes. Therefore, cells of the leaves will die. Now take a peel of the leaf and put it on a slide. Put a strong solution of sugar or salt on the peel of the leaf on the slide. Wait for a minute and observe it. During observation, it is noticed that plasmolysis did not occur. This means that plasmolysis did not occur in boiled leaf cell. Thus, we can infer from the experiment that only living cells can absorb water and undergo osmosis, whereas dead cells are not able to absorb water. Hence, plasmolysis cannot occur in dead cells. Cell wall permits the cells of the plants, fungi, and bacteria to tolerate hypotonic solution without bursting. In hypotonic solution, the cell absorbs lot of water and swell through the process of osmosis. Due to this, the cell swells, building pressure against the cell wall. The wall also exerts equal pressure against the swollen cell. This characteristic of plant cells help them to survive in dilute solution.
Nucleus.
Have you ever wondered why do we use iodine solution to prepare a temporary mount of onion peel? When we use the iodine solution, the contents of the cell, being of different composition, take up the solution and get colored. Similarly, if no solution is used to mount the cell, the components of the cell won't be clear. Iodine solution is an example of stain. Different stains like safranin solution or methylene blue solution are used. Due to the different chemical composition of cell components, some regions appear darker than other regions. Let us perform an activity to observe the cells in human body.
Activity. Take a clean glass slide with a drop of water. Using an ice cream spoon or cotton bud, gently scrape the inner surface of your cheek. The material which is slimy gets stuck on the spoon.
Transfer this material on a glass slide. Put a drop of methylene blue stain on the smear and put a cover slip.
When we observe the slide under the microscope, we can see small circular cells with dense cytoplasm. A darkly stained blue spot is observed in the center of each cell. This is the nucleus.
The cheek tissue shows many cells which are of similar type. This represents the structure of an animal cell.
The nucleus is a large spherical body. Usually, it is situated at the center of a cell. It is the most important part of the cell. It controls all the activities of a cell and takes part in cell division. It decides the function which a cell will carry out after maturation. The structure of a nucleus is clearly observed with the help of an electron microscope. It is covered by a double-layered nuclear membrane. The nuclear membrane has pores which allow the transfer of products from the inside of the nucleus to the outside, that is, the transfer of material from the nucleoplasm to the cytoplasm. The nucleoplasm contains one or more round-shaped nucleoli and a network of fibers called chromatin fibers. Nucleolus is not bounded by a membrane. Nucleolus is a site of ribosome formation, hence it is known as factory of ribosomes. Chromatin fibers are thin thread-like structures composed of DNA, that is, deoxyribonucleic acid and protein. These fibers get condensed to form short, thick chromosomes during cell division. Chromosomes are visible during cell division. DNA molecules transfer the hereditary information from one generation to the next. Functional segments of DNA are called genes. Usually, one gene controls one or more cell functions, but sometimes a single function is controlled by a set of genes. The nucleus acts as the control center for all the metabolic activities of the cell. It both regulates and coordinates all the life processes of the cell. If at any time the nucleus is removed from the cell, it ultimately dies. Besides its regulatory role, the nucleus also controls cell division. The nucleus contains factors or genes that determine heredity. It is also involved in transmission of hereditary characters from parents to offspring due to the chromosomes present in it.
In some organisms like bacteria and blue-green algae, nuclear region of the cell is not well developed due to the absence of the nuclear membrane. Such undefined nuclear region containing only nucleic acids is called a nucleoid. The cells which lack a nuclear membrane are called prokaryotic cells.
The term prokaryotic means the forms which were present before the nucleus was evolved. Greek word pro means before and karyon means nut or kernel. These are the organisms with primitive or primary type of nucleus. These are the cells without membrane-bound organelles. The prokaryotic cells are very simple. The cells having membrane-bound organelles are called eukaryotic cells. In Greek, eu means true and karyon means nut or kernel. Karyon also refers to the prominent nucleus of a cell. The cells possess a well-defined nucleus which has a prominent nuclear membrane, nucleolus and nucleoplasm.
Let us study some differences between prokaryotic and eukaryotic cells. The size of prokaryotic cells is usually small, ranging from 1 to 10 micrometer. But eukaryotic cells are generally large, with sizes ranging from 5 to 100 micrometer. The nucleus is absent or not well defined in prokaryotic cells, and the nuclear region or nucleoid is not surrounded by a nuclear membrane. In eukaryotic cells, the nucleus is present and the nuclear material is surrounded by nuclear membrane. Prokaryotes lack membrane-bound cell organelles, but eukaryotic cells have membrane-bound cell organelles like endoplasmic reticulum, Golgi body, mitochondria, plastids, lysosomes, peroxisomes, and vacuoles. In photosynthetic prokaryotic bacteria, the chlorophyll pigment for photosynthesis is associated with membranous vesicles, whereas in case of eukaryotes, the chlorophyll pigment is embedded inside the specialized structure called plastids. In prokaryotic cell, the chromosomes present are single, whereas in eukaryotes, the number of chromosomes are more than one.
Cytoplasm. When we observe the cells of onion peel, the cells are flat. They are full of fluid which lies between the plasma membrane and nucleus. This fluid matrix is called cytoplasm. The cytoplasm is living and is constantly moving. It is a jelly-like substance. Many cell organelles lie in the cytoplasm. Cellular chemical reactions occur in the cytoplasm. In animal cells, cytoplasm is more granular and dense, while in plant cells it is thin. It is mostly pushed to the periphery due to a larger central vacuole.
As prokaryotes do not show the presence of any nuclear membrane, membrane-bound cell organelles are also absent. On the other hand, the eukaryotic cells have well-developed nuclear membrane as well as membrane-enclosed organelles. Membranes are one of the most important part of the cell. This can be explained with the help of an example of a virus. a virus lacks membranes and do not show any characteristics of life until it enters a living body and multiplies inside the organism.
Endoplasmic reticulum, ER. Endoplasmic reticulum is the intracellular transport system of a cell. It is a large network of membrane-bound, fluid-filled, interconnecting tubules and sheaths. ER is continuous with the plasma membrane on the outer face and the nuclear membrane on the inside. Endoplasmic reticulum is of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, RER, looks rough under an electron microscope. It has particles called ribosomes attached to it. Ribosomes are present in all active cells. Ribosomes are the sites where proteins are synthesized. Rough ER helps to transport the proteins synthesized by ribosomes. The proteins are transported to that area of a cell where they are required. Smooth endoplasmic reticulum, SER, is without ribosomes. It is meant for the manufacture or synthesis of fat molecules or lipids. Endoplasmic reticulum acts as an intracellular transport system. It acts as a supportive framework for a cell. This is because it extends from the nuclear membrane to the plasma membrane. It synthesizes proteins and lipids or fats and transports them. Some lipids synthesized by ER are used as building blocks of plasma membrane.
The process of building of cell membrane with the help of lipids and proteins is called membrane biogenesis.
Some other proteins and lipids are used in the cell metabolism as enzymes and hormones. It transports proteins synthesized by ribosomes, that is, RER. SER of certain cells of liver plays an important role in defense mechanism of body. It has a crucial role in detoxifying many poisons and drugs which enter our body through food, air or water. The toxins are made water-soluble so that these water-soluble toxins may be excreted through urine. Golgi apparatus or Golgi complex.
Golgi apparatus was first discovered by a German scientist, Camillo Golgi.
Camillo Golgi, who discovered Golgi apparatus, was born in 1843. He studied medicine in the University of Pavia. He developed a staining technique known as black reaction. He explored the nervous system using this newly developed technique.
In 1906, he shared the Nobel Prize with Santiago Cajal for the work on structure of the nervous system.
The Golgi complex comprises of about 5 to 8 or more sacs which resemble deflated balloons. These sacs are placed approximately parallel to each other. These sacs are called as cisternae. They are filled with different enzymes.
Cisternae have connections with the membranes of endoplasmic reticulum and thus is one of the most important part of the cellular membrane system.
Golgi apparatus is considered as a packing and forwarding department for the chemicals produced by the cell. Golgi complex is the secretory organ of the cell. It modifies, sorts and packs materials synthesized in the cell. These materials include proteins, lipids, enzymes, pigments, mucus secretions, etc. It dispatches these materials to the various target organelles inside the cell, such as plasma membrane and lysosomes. Sometimes it dispatches materials outside the cell. It produces vacuoles and secretory vesicles. It plays the role in the formation of cell wall, plasma membrane, and lysosome.
Sometimes complex sugars are also synthesized from simple sugars in Golgi apparatus.
Lysosomes. Due to various metabolic activities of the cell, metabolic waste is produced. Lysosomes digest the waste. Lysosomes are simple, single membrane bound sacs. They are filled with digestive enzymes. They are capable of digesting and breaking down all organic matter. As observed in animal cells, lysosomes are the cell's waste disposal system. Lysosomes are absent in plant cells. The lysosomes represent the digestive system of the cell. The foreign material like bacteria and viruses which invade the cell are destroyed by the lysosomes. Thus, they protect the cell. They remove worn-out and poorly working cellular organelles. They remove all the organic debris. When a cell gets old or is damaged, lysosomes burst and the enzymes digest their own cells. Hence, they are also called suicide bags. Mitochondria. Mitochondria are the powerhouses of the cell. They produce the energy required for the cell. Under electron microscope, a mitochondrion is seen as a double membrane bound structure. The outer membrane is porous while the inner membrane is deeply folded. These folds are called as cristae. Due to these folds, the surface area of inner membrane is increased to a great extent. The larger surface area ensures the efficient functioning. The inner cavity is filled with proteinaceous gel-like matrix. The matrix contains ribosomes, phosphate granules, and DNA. Due to this, mitochondria are capable of making some of their own proteins. With the help of enzymes, the mitochondria oxidize carbohydrates and fats present in the cell. During this oxidation process, a large amount of energy is released. The energy released during the process is used by the mitochondria to produce energy-rich compound ATP, adenosine triphosphate. ATP is energy currency of a cell. There are numerous mitochondria in animal cells, whereas plant cells have fewer mitochondria. Mitochondria produce the energy-rich compound ATP, which is the energy currency of the cell. The organisms use the energy stored in ATP to synthesize chemical compounds like proteins, carbohydrates, and lipids. The body uses the energy stored in ATP to do mechanical work such as movement of muscles, conduction of nerve impulse, production of heat, and other such activities. Plastids. The leaves are green colored, the flowers shown in various colors, and so do the fruits. For example, raw mango or raw tomato is green in color, but ripe mango is yellow and ripe tomato is red in color. The organelles which are responsible for imparting this color are the plastids. Plastids are double membrane bound structures. They are present only in plant cells. There are two types of plastids. One, chromoplasts, that is colored plastids, and two, leucoplasts, colorless or white plastids. Chloroplasts are a type of chromoplasts which are green in color. They contain a green colored pigment called chlorophyll. Chloroplasts are important for photosynthesis in plants. These are energy factories of plant cells. That is, they manufacture the food by trapping solar energy. This light energy is converted to chemical energy. This energy is stored into the chemical bonds produced during synthesis of food. The food is in the form of carbohydrates, that is glucose. Food is stored in the form of starch. When we observe the structure of chloroplasts, it shows two distinct regions, stroma and grana. Stroma. It is the colorless proteinaceous matrix. It contains various photosynthetic enzymes, DNA, ribosomes and starch granules. Granum. It is stack of thylakoids. Thylakoids are discs containing chlorophyll and other photosynthetic pigments. The tubes joining grana are called intergranal lamellae. Due to presence of DNA and ribosomes, plastids can self-replicate like mitochondria. Chloroplasts help to harness solar energy and convert it into chemical energy in the form of food. Chromoplasts give colors to flowers and fruits. Leucoplasts are involved in the synthesis and storage of various kinds of food like starch, oils, and proteins. Vacuoles. Vacuoles are the storage sacs for solid or liquid contents. They do not have any basic shape or size. The structure of the vacuole changes according to the need of the cell. In animal cell, vacuoles are few, small in size and are of temporary nature. In plant cells, we come across one large vacuole known as a central vacuole or more vacuoles which are large in size. The membrane of the vacuole is single-layered.
Vacuole occupies almost 90% part of the cytoplasm in mature plant cell. In plant cell, vacuoles are full of cell sap. They provide turgidity and rigidity to them. It contains a variety of substances including carbohydrates, proteins, minerals, sugars, amino acids, pigments, and organic acids. Food vacuoles of single-celled organisms like amoeba and paramecium store food materials that the animal has consumed. Some unicellular organisms have specialized vacuoles which help in removing excess water and some waste from the cell. Each cell has its structure and function to perform. The ability of the cell to function properly is the result of the organization of its membrane and organelles present in a specific way. A cell has a basic structural organization which helps it to perform different functions like respiration, obtaining nutrition, forming new proteins, or expelling waste materials. Thus, it can be said that the cell is the fundamental structural and functional unit of all living organisms.
Let's do a quick check. Let's do a quick check. Let's do a quick check.